Hello everyone, this is GATE 2023 Life Sciences Section S Microbiology. Question number 71. Catabolite repression in bacteria is regulated by concentration of A. amino acid, B. glucose, C. messenger RNA and D. lactose. This is the NPTEL online course reference which explains the bacterial metabolism. So catabolite repression is observed in Lac-Operon model. Okay, Lac-Operon model, and this Lac-Operon model is very famous. It's found in E. coli cells. Okay, in E. coli cells we have Lac-Operon model. So I'll explain you this Lac-Operon model and also catabolite repression. Okay, so this is the Lac-Operon model. So as the name suggests, Lac-Operon, operon basically means cluster of genes. So these cluster of genes are responsible for the lactose metabolism. Okay, lactose is a sugar made up of glucose and galactose. Okay, these are the two simple sugars. It is made up of two simple sugars, glucose and galactose. And the Lac-Operon consists of three genes, LAC-Z, Y and A. So LAC-Z gene is mainly responsible for breaking lactose into small basic sugars like glucose and galactose. Whereas LAC-Y and LAC-A gene is responsible for lactose permeabilization and absorption in the bacterium. Okay. Apart from these three genes, there are upstream of these genes we have a regulatory region. So this yellow color is where the repressor protein binds. So this is the repressor protein here in the red. So I'll explain you what is this repressor protein. This is the site of RNA polymerase binding. So this RNA polymerase will transcribe the Lac-Operon genes. Okay, so this is the direction of transcription, right? So RNA polymerase will transcribe this Lac-Operon genes, and this blue-green color region here, it is a very important regulatory site known as CAP site, catabolite activatory protein site, also known as CAP site. Okay, and we have LAC-I gene. Okay, so this LAC-I gene produces mRNA and produces repressor protein. This red repressor protein is produced by this LAC-I gene. Okay, and this LAC-I gene will go and fit here in the yellow region. This is the binding site of a repressor protein. Okay, and it will not allow RNA polymerase to transcribe these genes. Okay, so what happens in presence of lactose? So whenever bacteria senses lactose in the medium, so this lactose is converted into allolactose. Okay, this allolactose is an isomer of lactose. So whenever bacteria senses lactose in the medium, it is converted to allolactose, and this allolactose will bind in this repressor protein. It has a site in the repressor protein, binding site. It will bind and it will inactivate the repressor. Okay, now since the repressor is inactivated, it will no longer bind to this Lac-Operon region here, and the RNA polymerase now is free to transcribe Lac-Operon genes. Okay, and then we'll have metabolism of lactose. Okay, so in presence of lactose we have inactivated repressor protein, and then we'll get transcription of LAC-Z-Y-A gene. Okay, so this is in presence of lactose. So this is in presence of lactose. First condition, in presence of lactose, we'll have transcription of LAC-Z-Y-A genes. Okay, but this is not as simple as that. Only presence of lactose is not sufficient to to transcribe these genes, because this RNA polymerase is very weak. It has a weak binding. It has a very weak binding to the lac-operon gene here, to the binding site, and it falls off and it produces very few amount of so the expression levels are very less. Expression levels of these genes will be very less in only presence of this RNA polymerase. Okay, the binding is very weak, and hence we have very low expression of these genes in presence of lactose. So what will happen when we provide both glucose and galactose? So bacteria are very clever. If they already have glucose, so glucose is a basic sugar for the bacteria, and the metabolism of glucose, the genes required for the metabolism of glucose are already present inside the bacteria. They are the housekeeping genes, and thus they are already present inside the bacteria. But genes for lactose metabolisms are inside this lac-operon, right? And bacteria will not utilize its energy to produce these genes to metabolize lactose in presence of glucose. If it is getting simple sugar glucose, okay, first it will utilize glucose, it will grow, and after utilizing all the glucose from the medium, then it will shift to lactose. Okay, so this phenomena in which the bacterium E. coli prefers glucose over lactose in presence of both glucose and lactose in the medium is known as catabolite repression. Okay, catabolite repression. So how it is exactly preferring glucose? So what happens when there is already glucose in the medium, right? Glucose, which is a much simpler sugar than lactose, it is all present in the medium. It is utilized by the cell to give us more ATP. Okay, so we'll have higher levels of ATP inside E. coli. And we also know that ATP levels inside the cells are balanced with that of cyclic AMP levels. Okay, so whenever ATP levels are high, cyclic AMP levels will be low, and vice versa. Okay, when cyclic AMP levels are high, ATP levels will be low. So in presence of glucose, we have glycolysis pathway, and from glycolysis pathway we have generation of lots of ATP molecules. Okay, so inside the cell adenosine molecules, okay, they are constant, and those constant levels are divided among ATP and cyclic AMP. So intuitively we know that when ATP will be higher, cyclic AMP will be lower. Okay, so overall adenosine levels will be constant inside the cell, and these constant levels always change depending upon either ATP is whether ATP is in higher concentration and whether cyclic AMP is in higher concentration. Okay, so so what happens when cyclic AMP is in lower concentration? This cyclic AMP is a very important catabolite repressor. Okay, it is a catabolite repressor. What it does, it goes and binds to this CAP site, catabolite repressor protein site. So cyclic AMP will come and bind to this site. Okay, CAP site here. I told you that this is very important regulatory unit of lac-operon. The cyclic AMP units, it will come and bind to just upstream of the RNA polymerase. Okay, and this binding, okay, this binding will increase the binding of RNA polymerase, and it will be a strong transcription. The transcription will be strong, and there will be high expression high expression of LAC-ZYA genes. Okay, because this polymerase will have a strong binding, and this binding is much influenced by cyclic AMP, by binding of cyclic AMP to the CAP site, catabolite repressor protein site, okay, on this lacoperon. So when this cyclic AMP will come and bind, only when the glucose levels are very low. When the glucose levels are low, ATP levels will also go down. Okay, if ATP levels are gone down, the cyclic AMP levels will increase. Okay, so this is the catabolite repression. Okay, this is catabolite repression. So inside E. coli, there are 3 different possibilities of glucose and lactose metabolism. So in first case, we have only glucose. There is already high amount of glucose present in the medium, so we'll have high amount of ATPs. Okay, high amount of ATPs, so the cyclic AMP levels will go down. If cyclic AMP levels are going down, what will happen? There are 2 things it will happen. First, there won't be a protein bound here which will stabilize RNA polymerase, and there will be also an active repressor protein, right? This active repressor protein will come and bind here because there is no lactose, hence there will be no allolactose also, and this repressor protein will repress this RNA polymerase, and it will inhibit the transcription of these genes. Okay, so in presence of glucose, lacoperon is shut down, right? In presence of only glucose, lacoperon is shut down. Suppose we have glucose in B type of in B case, we have glucose and lactose. What will happen? So the cell will first utilize the glucose and it will repress lacoperon, and this process is known as catabolite repression. So in presence of glucose, again there will be high ATPs. Okay, there will be high amount of ATP, and the cyclic AMP levels will go down, and the cyclic AMP will now not be able to activate this catabolite repressor protein site on the lacoperon, and the weak RNA polymerization will lead to a lower expression. Okay, so this RNA polymerase will lead to lower expression of genes. Okay, so and we have lactose also. So lactose will convert it to allolactose, and this inactive repressor, thus there will be lower expression, right? And once all the glucose is over and we are now left with only lactose, or we are given E. coli only lactose, then what will happen? There will be, since there is no glucose, there is no ATP production in the cell right now. There is less ATP, and the cyclic AMP levels will increase. These cyclic AMP levels will first go and bind to this CAP site. This will further increase our binding of RNA polymerase, and we'll have higher expression of LAC-ZYA gene. Okay, and also in presence of lactose, this repressor will become inactive and will get production of LAC-ZYA gene. So coming back to our questions, amino acids, none of the amino acids play any role. Messenger RNA, no, there is no as such any specific mRNA here. Glucose is involved, yes. Lactose is also involved, but catabolite repression, this phenomena is only observed in presence of glucose, right? In presence of glucose, there is catabolite repression of lactose, right? Lactose related genes. So in presence of glucose, we have catabolite repression of LAC-ZYA genes in lac operon. Okay, so lactose is not playing any role even in presence of lactose. Or absence of lactose. Okay, the glucose is the regulatory molecule here. The glucose will maintain the ATP and cyclic AMP levels, right? It will balance out the ATP and cyclic AMP levels, which are the key regulators of this catabolite repression in lac opera. Okay, thank you. So the answer is B, glucose.